Our last step we got, I've transcribed a whole bunch of it, right? I've multiplied it. Okay, I've done all that, figured out how to do that, saw how that happens. Okay, so let's say I want to copy a DNA segment to go out and make a protein. Well, you transcribe it. You transcribe it, that takes place in the cytoplasm, or I'm sorry, in the nucleus, in the nucleoplasm. Then I go through a number of various different steps where I copy one segment, one side of the DNA helix, right? Put a cap, put a tail, splice out the bad, put the exons together, and ta-da, now it can leave through those nuclear pores and head out into the cytoplasm. Now what does it do? Now we're out in the cytoplasm, okay, in the cytoplasm, okay. And now I can read the transcript and I can translate that. I could translate that mRNA segment. And that helps to give me a protein. I copied it, transcribing it. Then I read it. And when you read, you translate. And that allows me to make a protein. Reading the amino acid sequence. And remember, amino acids strung together get you a protein. This happens at a ribosome. So ribosomes are out within the cytoplasm. So this occurs at a ribosome. Okay? It doesn't happen willy-nilly in the middle of nowhere. There's got to be a ribosome attached to get this thing to happen. This is where we see our tRNA molecule. Our RNA is, remember, is the ribosomal subunits. Okay? But it's really that tRNA that's doing a lot of the heavy lifting here. It has to grab a hold of that amino acid and bring it here. Okay? And when I say bring it here, bring it to the ribosome. Okay. Oh, we have a portion here of the tRNA molecule that actually binds with the ribosomal RNA. Well, this right here binds with mRNA found within the ribosome. And we'll see how that just fits here in a second. Okay. And the other end of the tRNA molecule binds to the amino acid. Amino acid. Okay. So there's your tRNA molecule. It's actually more complex. I mean, you can see it looks really complex. It's not as simple looking as that. But let's just keep it simple, right? Let's just keep it simple. You can see there's a while. I don't really care for you to know that. The book goes into it. It's, you know. There's a small degree of variation. I should have gotten rid of it because I'm not going to get into it. The book does if you want to read about it. There's a little bit of wobble that can happen, so it can change. So what are the steps of translation? Okay, transcription is pretty easy. RNA polymerase, unzip, copy, cap, tail, splice, go. For translation, I have to translate. I have to read that mRNA strand. Okay. So as we're going to see here in a second, this mRNA strand actually feeds itself through a ribosome. So it's actually going to associate with the ribosomal RNA. Okay, this could be found in the cytoplasm, floating by itself. This could be found within the cytoplasm, but attached to the rough endoplasmic reticulum. And remember, that ribosome is what makes the protein. It reads the information, okay? Hence the name translate. Okay? And when it reads it, that ribosome is basically going to make an announcement that the tRNA picks up that says, yo, I need you to bring uracil. I need you to bring threonine, methionine, okay? I need you to bring the specific amino acid, okay? And tRNA brings in the specific amino acid. Okay. And then through a process that happens within the ribosomal subunits, between the ribosomal subunits, we connect these individual amino acids with a peptide bond, and we just continue to add peptide bonds, and these amino acids get longer and longer and longer, and that's our primary structure of a protein. Just the reading, the translating that occurs at the ribosome. We have 64 different kinds, I mean, they each kind of carry one of the 64 different possible, anticodons. Okay. We have the codon that's on the mRNA molecule. We have the anticodon on the tRNA molecule. Remember, complementary base pairing, that's why. So if the mRNA says AUG, then the tRNA is going to have UAC, okay, the complementary base pair. But it's specific, okay? They still code for the same thing, bringing in the same thing. tRNA synthase attaches, okay, yeah, I mean, there's another enzyme, I don't really care. I'll just put a cross through it. All TNA molecules with a specific, yeah. Okay. So here's my codon, CUU, and there's my anticodon, GAA. Pretty simple, pretty straightforward. All right, so when I look at this ribosomal subunit, it has basically 3 sites. The EPA, okay, the exit site, the peptide site, and the amino acid site, okay? The amino acid site is going to go into A. As it moves down, A goes to P. Well, that leaves A open. So another amino acid is going to get dumped into the A. When that happens, the amino acid at the P site and the amino acid at the A site, they get to connect through a peptide bond. At the next site, it's called the exit site, and you could probably imagine what happens when you get to the exit site. You get to leave. Okay. Let's see what that looks like. Okay. So here's my large and my small ribosomal subunit, right? And we talked about how mRNA kind of gets fed right between them and gets read. Okay. So here's my EPA site. Okay. Here you can see the incoming tRNA molecule adds to the A site. Amino acid. Okay. Here, okay, I have an amino acid that came in right before it. It just moved down. This one here. As you can kind of see, through a peptide bond, I'm going to attach that amino acid to the growing chain here. There it is. Okay. So now this guy is devoid of an amino acid on its one end. As it advances, it goes to the exit site and it exits, hence the name. This one was here at the A, and now it's there at the P site. Well, the A site's open, so we're going to bring in another because I haven't gotten to a stop codon yet. And the same thing is going to happen, and this continues. And this can happen at multiple spots along the length of the mRNA molecule. As long as there's a start codon there, it can happen. So translation, we basically break up into 3 steps. And what I just kind of talked about there, I mean, these are the 3 steps, okay? Step 1 of translation, the initiation, okay? Starts with the UAC codon, okay? AUG, methionine, Okay, it always starts with the start codon. And the anti-codon of the codon is UAC. And it can actually bind to the specific P site, but who cares? Okay, it enters into the A and then just moves right over to the P. Just look at it like that. Okay. Again, what do we need? We needed a small and we needed a large ribosomal subunit, right? Large and small. Yes, doctor. We need the mRNA transcript. Yes. Okay, yes. There it is. See? There it is. And we need the initiator tRNA. Okay, UAC. Okay, methionine. Remember, AUG was methionine. UAC is the anticodon. So we need the starter. We need the initiator. Right there, start codon. Start codon. Okay. So as long as we got the ribosome, as long as we got the mRNA, and as long as we got the initiator, we're good to go. So we start with the initiator anticodon. Pops into the A, moves over to the P. As it moves over to that P site, active site, or I'm sorry, the amino acid site is wide open. So whatever codon this is, I can find the anticodon, it'll bring with it, and it'll bring with whatever amino acid it is. Okay. There's the anticodon. There's the codon. The anticodon will be C, U, G. There you go. CUG. All right. And the peptide bond gets formed, and this one will go to the exit. This is exit, and this guy will get jetted off. Boom, there it goes. It continues to go, and we continue to grow this chain of amino acids. As this peptide bond is forming, and as this chain of amino acids is growing, it is shockingly called step 2, elongation. Okay. That second tRNA that came in moves to the P site. The initiator that started it all moves to the E site and exits. The ribosome reads the next codon, tells the tRNA, I need this anticodon, please, over here. It brings in, binds to the A site. From the peptide bond, everybody moves down the line, and the amino acid sequence gets longer and longer. Eventually it stops. And the third step, excuse me, and the third step of translation is shockingly called termination. It'd be more prudent if it was just called stop, right? But it's called termination. All right. And this only occurs if we get that stop codon, right? We had 3 of them. I'm not going to really probably ask the 3 stop codons. I'll probably ask what the potential start codon is because there's only one of those. But any one of those stop codons, as soon as it pops into the A, we have a peptide bond and boom, it stops. It basically is coding for a release factor. And when that release factor binds with the stop codon, that's what it's bringing, a release factor. It's a protein that says, jettison, go forth, and conquer. Okay. So here's that stop codon. There's the release factor, and that causes the ribosome to disassociate. That causes that amino acid sequence to detach and go to wherever it needs to go. It can stay inside the cell. It can be packaged up and removed from the cell. It can be made into whatever in the world the cell needed it for. And this just kind of puts it all together.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, one that codes for start, and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons.
Sorry. This ability to understand what a chromosome is, what a chromosome looks like, how to isolate that chromosome, how to isolate that gene, allows us to really understand and pick and choose how to control what kind of product, what kind of protein we can ultimately get a cell to do or a cell to make. And by being able to control that, I can fix defective cells, which sounds fantastic. And that's the basis for what we're going to be talking about in chapter 14. We're talking about cloning, and we're talking about gene manipulation. And we've been doing this since, well, in terms of crops for agriculture business since like the 70s. Being able to do it in animals has been something a little bit more difficult until the 90s. And now we've manipulated several things. So being able to know how to understand where we came from with our understanding of what codes for stuff in the next generation, being able to isolate that, see it, understand it, see it actually moving from one generation to the next, or from one species or one virus into another bacteria, seeing how that can now be replicated in that bacterial line, being able to isolate that, understand how it is copied in a cell, whether it be pro or eukaryotic, and then understanding the process of copying a specific segment, because I don't need the whole damn thing copied. I usually just need a specific protein or two made. Okay, let's say we're fighting off an immune response. Okay, we're fighting off a foreign invader. I just need to make some proteins that code for inflammation. to get a white blood cell to come inside me. So I can now fight off that foreign infection. Okay, so I just need a couple segments. So I can understanding how that isolates, how it gets read and spliced together to become mature. And then when it leaves the nucleus and goes out into the cytoplasm to be translated, how that process occurs. So I can make a specific amino acid sequence to code for a specific protein, to code for a specific thing to occur within a cell, or to be gotten rid of, to occur in another cell somewhere else, enter into the blood. That has opened Pandora's box into what is biotechnology in genetic manipulation. Because I have a bunch of videos in the next one that talk about, the beauty behind the science of it and our understanding, our ability to fight off disease and, prevent diseases and really bad just genetic conditions from occurring. Yes, but what's the specter behind the corner? Where does it stop? Yes, we have laws here in the United States that, regulate and prevent us from, but not every country is the United States with a moral compass. And in chapter 14, we'll talk about the biotechnology and the application of what we discussed here today in this central dogma of biology. I hope you guys enjoyed it. It's pretty interesting. I know. Okay. Going over it a few times. All right. Will make it a much more easier digestibility for you to be able to, get it, understand it, and be able to talk about it a little bit on the exam. On this exam, I'm probably not going to go in and do a hard count on did you get all the little specifics, but the general understanding of what's going on, that's what you should be able to get from this. See you next time.